My name is Diane-Marie Smith, and I'm here to talk to you about some research I did for Miami-Dade College examining enzyme replacement therapy using human neural progenitor cell model of CLN2 disease. CLN2 is a autosomal recessive neurodegenerative lysosomal storage disorder called ceroid neuronal lipofuscinosis type 2. This is a genetic mutation that's inherited, and this genetic mutation causes a deficiency in liposomal enzyme tripeptidyl peptidase. This enzyme is directly responsible for disposing waste in the cells. Cells that are primarily impacted by this disease are in the brain, and over time, since the cells in the brain cannot remove waste, the waste starts to accumulate and the cells rupture and die. They lose function, and it is a very bad disease. You have a child that's usually developing normally up until around 3. Then they have a rapid decline in their motor function, in their language. They start to have seizures, and they typically don't live past 8 to 10 years of age. Currently, the only treatment is kind of crude. It's called Brineura, and it is delivered to the brain via intracerebral ventricular infusion. This basically means that they install a catheter in the brain of the child, and once every 2 weeks, the TPP1 is flooded into the brain and diffuses via concentration gradient. It cycles through the ventricles. You can kind of see there with my mouse, and it gets distributed through the central nervous system. Where this can cause issues, and the questions that we have in research is, the ventricles are home to the subventricular zone, and this zone is crucial in how cells proliferate in the brain. It's the home of neural stem cells, and these stem cells go on to become neurons, astrocytes, and oligodendrocytes. So we asked, how does different concentration gradients of TPP1 affect how they grow, divide, die, proliferate, etc. So we took normal cells, let them grow as they usually do, and we analyzed them in a microscope and counted them to see how they grow, and then we're going to treat them with different concentrations of TPP1 to see if there's any changes, and if so, at what point are the changes, and this way we can figure out what is too much TPP1 to deliver into the brain, what is too little. My biggest contribution to this project was finding this program, Cell Profiler. Cell Profiler is an open source program by the Broad Institute. It is a cell counting software that you can give certain commands to have it specifically find different types of things in the cell, and then it will count it for you. Here you have an example of some of the cells. These were normal cells. They were not treated with TPP1. And so you have your human neural progenitor cells, and they were treated with the red parts here are expressing double cortin, which means that they basically turn into oligodendrocytes. Oligodendrocytes have many processes, up to 50 many times. They kind of stretch out in the mind and provide scaffolding and insulation for different, for really everything in the brain. Here you have, so the trick is, and these are all pictures of the same slide analyzed through Cell Profiler. All of these little gray dots that you see in the upper left are DAPI stained cells. They glow blue. They're in grayscale here for the program to analyze it and give you this pretty rainbow image here. The DAPI is a universal stain, so it'll stain every nucleus that's on the slide. And down here in the bottom, you have your double cortin expressing processes for the oligodendrocytes. Now, when you look and see, here's a closer view, it looks like there's, all of the nuclei have processes, but really they don't. It's only these really bright spots where they're originating from. So there's only in these slides 18 cells, which the program counted here. You can kind of see in green, and how to kind of come up with clever ways to have it target those areas. And that's how we get our numbers. We're still continuing to do this experiments and treatments of TPP1, and we have yet to analyze those slides, but hopefully soon I'll have lots and lots of more data that I can analyze with this program.